few moments later.
Now is an introduction. Blood is a fluid that requires carbon dioxide and other waste materials out of the body while supplying oxygen and nutrients to the cells. In technical terms, blood is a transport liquid that is pumped to every region of the body by the heart or similar structures and then returned to the heart to be repeated. Artificial blood is a potential substitute blood product that is capable of performing blood's most essential job, which is to deliver carbon dioxide and oxygen. In situations where there is a significant blood loss due to major disasters, artificial blood may be a life-saving treatment. However, the products now under development are unable to perform secondary tasks of blood, such as infection prevention. Now, I will tell you about background and history about artificial blood. A significant breakthrough in the development of artificial blood came in 183 with the creation of a Ringer solution. So, Ringer solution is a solution composed of sodium, potassium, and calcium salts. In research using part of a frog heart, scientists found that the heart could be kept beating by applying the solution. This eventually led to finding that the reduction in blood pressure caused by a loss of blood volume could be restored by using Ringer solution. This product evolved into a human product when Loctet was added. While it is still used today as a blood volume expander, Ringer's solution does not replace the action of red blood cells, so it is not a true blood substitute. Okay, let's look at characteristics of artificial blood. Artificial blood is safe to use and compatible for human body. Artificial blood also able to transport and release oxygen where needed. Artificial blood storable and durable for a longer time period. Free of pathogen and toxins. Artificial blood viscosity similar to blood. And then artificial blood is low cost to make.
Next, material for making the artificial blood. The ideal artificial blood product has the following characteristics. First, it must be safe to use and compatible within the human's body. That means the difference of blood type should not matter when an artificial blood is used. It also means that artificial blood can be processed to remove all disease-causing agents such as virus and organisms. Second, it must be able to transport oxygen throughout the body and release it where it is needed. Third, it must be shelf-stable. Unlike donated blood, artificial blood can be stored for over a year or more. This is in contrast to natural blood, which can only be stored for 1 month before its breakdown. There are two significantly different products that are under development as blood substitutes. They differ primarily in the way that they carry oxygen. One is based on perfluorocarbon, which is PFC, while the other is a hemoglobin-based product. Perfluorocarbons or PFC. As suggested, PFC are biologically inert materials that can dissolve about 50 times more oxygen than blood plasma. They are relatively inexpensive to produce and can be made devoid of any biological materials. This eliminates the real possibility of spreading an infectious disease via a blood transfusion. From a technological standpoint, they have two significant hurdles to overcome before they can be utilized as artificial blood. First, they are not soluble in water, which means to get them to work, they must be combined with emulsifier. fatty compounds called lipids that are able to suspend tiny particles of perfluorochemical in the blood. Second, they have the ability to carry much less oxygen than hemoglobin-based product. This means that significantly more PFC must be used. One product of this type has been approved for use by Federal Drugs Administration (FDA), but it has not been commercially successful because the amount needed to provide a benefit is too high. Improved PFC emulsions are being developed, but have yet to reach the market.
Now, I'm Nafisa and I'm gonna talk about the hemoglobin-based product. So, what is hemoglobin-based product? Hemoglobin-based product is basically the same as our real hemoglobin in the red blood cell that have the ability to carry oxygen throughout all the body. However, it is a little different than the real blood because it doesn't contain in a membrane, so the blood typing is eliminated. Blood typing is a method to determine what type of blood are you. Hemoglobin can either be obtained by removing it from the expired human blood cells or cow blood cells, and also by chemically modified process into a product. However, raw hemoglobin can be broken down into smaller molecules and become toxic in our body. It is because, as I mentioned earlier, the hemoglobin doesn't contain in a membrane, so it becomes a free molecules within our body. In order to overcome this problem, chemically cross-linking molecule and also recombinant of DNA technology is used to produce a modified protein and also to stabilize the hemoglobin. Stabilized hemoglobin solution, polymerized hemoglobin solution, and also conjugated hemoglobin solution. These are the examples of the manufactured hemoglobin through DNA recombinant technology and also chemically cross-linking.
Next, for raw materials, different raw ingredients are used depending on the type of artificial blood that is produced. Both spirited hemoglobin and synthetically hemoglobin can be used in hemoglobin-based product. Amino acid containing substance are used by manufacturers to synthesize hemoglobin. These are the substances that are utilized by both plants and animals to make the proteins necessary for life. To make hemoglobin, 20 naturally available amino acids can be utilized. Certain chemicals properties are shared by all amino acid molecules. They consist of a side chain, carboxyl groups, and amino groups. The different amino acids are distinguished by the makeup of their side chain. A particular kind of bacteria and all the supplies required to cultivate it are also necessary for hemoglobin formation. Warm water, molasses, glucose, alcohols, urea, acetic acid, and liquid ammonia are examples for this. Hemoglobin is separated from human blood, are used in different kinds of artificial blood based on hemoglobin. Usually, donated blood that has expired before usage is utilized to get it. Hemoglobin can also be obtained from used animals' blood because this hemoglobin differs somewhat from human hemoglobin. It needs to be altered before usage. Next, we go for the manufacturing process. The production of artificial blood can be done in a variety of ways. For hemoglobin-based products, this involves isolation or synthesization of hemoglobin, molecular modification, then reconstitution in an artificial blood formula. PFC product involve a polymerization reaction, a method for the production of a synthetic hemoglobin-based product is outlined below. First, as you can see, to obtain hemoglobin, a strain of Escherichia coli bacteria bacteria that has the ability to produce human hemoglobin is used. Over the course of about 3 days, the protein is harvested and the bacteria are destroyed. To start the fermentation process, a sample of the pure bacteria culture is transferred to a test tube that contains all the nutrients necessary for growth. This initial inoculation causes a bacteria to multiply. When the population is great enough, they are transferred to a seed tank. Second, a seed tank is a large stainless steel kettle that provides an ideal environment for growing bacteria. It is filled with warm water, food, and ammonia source, which are all required for the production of hemoglobin. Other growth factors such as vitamins, amino acid, and minor nutrients are also added. The bacterial solution inside the tank is constantly battered with compressed air and mixed to keep it moving. When enough time has passed, the content of the seed tank is pumped to the fermentation tank. Third, the fermentation tank is a large version of the seed tank. It is also filled with a growth media needed for the bacteria to grow and produce hemoglobin. Since pH control is vital for optimal growth, ammonia water is added to the tank as necessary. When enough hemoglobin has been produced, the tank is emptied so isolation can begin. Fourth, isolation begins with a centrifugal separator that isolates much of the hemoglobin. It can be further segregated and purified using fractional distillation. This standard column separation method is based on the principle of boiling a liquid to separate one or more components and utilize vertical structure called fractionating columns. From this column, the hemoglobin is transferred to a final processing tank. For final processing, here it is mixed with water and other electrolytes, as you can see from the picture. The artificial blood can then be pasteurized and put into appropriate packaging. The quality of compound is checked regularly during the entire process. Particularly importance of frequent check made on the bacterial culture. Also, various physical and chemical properties of the finished product are checked, such as a pH, melting point, moisture contents, and others. This method of production has been shown to be able to produce batches as large as 2,640 gal. Once fermented, the hemoglobin is purified and then mixed with water and other electrolytes to create usable artificial blood.
Okay, now we are going to talk about effectiveness of artificial blood than the real blood. We are going to talk about prevention or complication of real blood transfusion, such as donor blood must match the blood type of the recipient. Then immune reaction to the blood, risk of infection. Developmental artificial blood product can be freeze-dried and kept for up to 2 years at room temperature. Donor blood must be tested for hepatitis, the human immunodeficiency virus (HIV), and other diseases, raising the cost of transfusion. Even though the tests are accidentally sophisticated, the danger of infection cannot be completely eliminated. Okay, then we are going to talk about blood substitute benefit. People with blood disorder who need regular long-term blood transfusion, such as patients with myelodysplastic syndrome, which is a kind of cancer that prevents immature red cells in the bone marrow from maturing. And then blood substitute may be used to preserve donor organs better and prevent reperfusion injury, which is organ tissue injury from return of blood supply. And then aplastic anemia, which is the bone marrow does not produce enough new blood cells. And then deliver oxygen faster than the real blood during an emergency and may minimize injury to the tissue, particularly during a heart attack.
My name is Nuri Nabila. So today I would like to tell you about the pros and cons artificial blood. So firstly, the pros is the artificial blood does not need to be refrigerated. Secondly, no need for blood matching. And lastly, accessible to patients in rural areas. For the cons, the artificial blood is expensive, does not mix well with the blood, and can cause unpleasant side effects. And lastly, does not carry as much oxygen.
In conclusion, we're going to talk about the effectiveness of the artificial blood. Based on recent studies, the rate of failure is higher than the rate of success. However, some of the manufactured artificial blood are accepted to be used as a donor organ preservation only, and no artificial blood they are accepted to be used for blood transfusion. It is because the research is still ongoing, and the problem faced for the researchers are the side effect of the artificial blood. Once it enters the human body, it can cause a life-threatening side effects such as hypertension, acute renal failure, and also myocardial infarction. It is due to the molecules used to produce the artificial blood. It cannot be accepted by the human body, so it end up become toxic compounds within the body and cause the side effect. However, once the research is full success, the artificial blood can be used at its full advantages in the medical field. It can help a lot of people that are having trouble with a blood transfusion because of the blood type, and also the lifespan of the artificial blood is longer than the real blood. It can be preserved for about a year compared to a real blood only for a few months. Okay, that's all for us. Thank you.